Hello, everyone. I am Marco Ruggiero, although, as you might have guessed, this is not my own voice, but that of a certain Ryan, lent to me by AI, which I am using for reasons of clarity. Today, I want to share the results of a very recent scientific research that is revolutionizing the fields of biology and medicine. These results are so extraordinary that if, a few years ago when I was teaching molecular biology at the university, a student had told me what I am about to tell you, namely that proteins create DNA in their own image and likeness, I would certainly have failed them. Such a statement would have gone against what has been defined as the central dogma of biology, ever since Watson and Crick discovered the role of DNA, the code of life, in 1953. In recent days, we have discovered that understanding the deep mechanisms regulating microscopic life often yields surprises that challenge the pillars of classical molecular biology, those we considered immutable truths for decades. Traditionally, we have always imagined the cell as a construction site with a central blueprint, the DNA. This blueprint is consulted to build operational structures, which are the proteins. In this rigid scheme, information flows in only one direction, from the genetic code of DNA to RNA, and from there to the proteins. However, a recent discovery shows us a reality that is much more dynamic and fascinating, where the order of things can be inverted, revealing a biological flexibility that, until recently, would have seemed like pure speculation. Imagine for a moment a skilled craftsman capable of building his own tools, not by following a written manual, but by using the very shape of his hands as a mold. This is exactly what happens with some recently discovered bacterial enzymes. Instead of reading a sequence of genetic instructions to produce new DNA, these enzymes use their own physical structure, their very protein conformation, as a guide to create new genetic material. This is a massive conceptual leap. It is no longer the gene that determines the function, but the protein itself that writes the code. Farewell, central dogma of biology. This phenomenon represents a kind of creative autonomy for the bacterial cell, demonstrating survival strategies that are extremely sophisticated and independent of classical schemes. This extraordinary biochemical capacity finds a profound resonance when we observe the unique complexity and biodiversity of Bravo. In such a rich and varied microbial ecosystem, the cooperation between different strains is not a random event, but the result of refined natural engineering superior to any genetic engineering humans could dream of. The bacteria composing this consortium are not simple passive spectators of our internal environment, but true guardians of health using cutting-edge defense strategies. The ability to generate DNA fragments starting from their own proteins allows them to prepare rapid and effective defenses against external threats, acting as a highly specialized militia that does not need to wait for central orders to take action. Employing this templateless DNA synthesis strategy allows the bacteria in Bravo to maintain extraordinary resilience. By defending themselves in such an innovative way, they ensure the stability of the entire microbial ecosystem. We know that the balance of our microbiome is fragile and constantly tested, yet it is precisely thanks to these exotic self-defense mechanisms that biodiversity is preserved. When good bacteria are able to protect themselves effectively, they prevent the proliferation of disruptive elements, ensuring that the microscopic garden we host remains flourishing and balanced. In conclusion, what science is bringing to light is a vision of bacterial life that is much more intelligent and proactive than once thought. The presence of such mechanisms within the complex Bravo system highlights the importance of approaches that respect and value natural biodiversity. In this scenario, the ability of a protein to generate life and protection without following a predefined manual is not just a laboratory curiosity. It is a testament to the strength with which bacteria maintain the harmony and balance of our microbiome, incessantly protecting our biological integrity.